Okay, our next speaker is Daniel Dever. He's a research instructor in the Department of Pediatrics, Division of Stem Cell Transplantation and Regenerative Medicine, and he is a recipient of an MCHRI postdoctoral support fellowship.
Thanks for the introduction. So I'm going to be talking today about a shift in a paradigm in stem cell transplantation where we correct dysfunctional genes in autologous hematopoietic stem cells to cure genetic disorders. So the one I'm talking about today is sickle cell disease. So sickle cell disease is caused by a single nucleotide mutation in the beta globin gene. And so this results from a transversion from an A to a T at the codon 6 of hemoglobin. And this results from a glutamate to a valine, which under deoxygenated conditions results in this polymerization of the hemoglobin molecule, and then thus its sickling shape, and then of course the name of the disease. There's over 3 billion DNA nucleotides in the human genome, and this disease results from one of these changes. The average lifespan is in the US is about mid-40s, but in Africa, about 50% of the children who have it die before their 5th birthday. So the only definitive and curative cure for sickle cell disease, I'd say at this moment, is HLA-matched hematopoietic stem cell transplantation. So there was this really great meta-analysis paper that came out showing the different sources of the stem cell transplantation, whether it be bone marrow, peripheral blood, or cord blood, but the overall survival is over 70%. But one of the main problems here is about only 10 to 15% of the patients have a suitable matched donor. And there can be complications of graft versus host disease and post-transplant immune dysfunction. One really interesting point from these studies was that a mixed chimerism showing that about 20% of donor chimerism can lead to a sustainable red blood cell compartment in these people. So with these nice stick figures showing this is kind of the old paradigm of the definitive and curative medicine for stem cell transplantation. So you have a donor about 15 to 20% of the time where your immune system matches the patient's immune system, and you undergo an allogeneic bone marrow transplantation to hopefully lead to a sustainable cure for the patient. What we're trying to do in the Porteus lab is kind of flip this paradigm to where you can take your own stem cells, so you always have a suitable matched donor in every single time, and you can correct the autologous stem cells leading to retransplantation, and it will avoid all of these complications shown here. And this all comes from the foundation of the hematopoietic tree that was briefly discussed by Sean Morrison. And so these hematopoietic stem cell and progenitor cells, these HSPCs, can, starting from the hematopoietic stem cell, can lead to this differentiated lineage to all the functional cells within your immune system. And this has been shown to be able to cure genetic diseases such as HIV, the primary immunodeficiencies, as I mentioned, sickle cell disease, and Wiskott-Aldrich syndrome. And so what we're trying to do is, in the case of sickle cell disease, to be able to fix these defective red blood cells, we have to be able to fix the gene in the repopulating self-renewing hematopoietic stem cells. And I'm going to show you a little data on that topic. And so how we do this is the last couple of years we've developed, I think, a platform, Biotechnology, for gene editing hematopoietic stem cells. The first discovery was made by I.L. Hendel and Rasmus Back, postdocs within the lab. And essentially what they did is they made chemical modifications to the termini of the guide RNA to make them more stable and resistant to exonucleases. And then Rasmus and I developed a platform using the AAV6 donor to be able to be a suitable and high-efficiency donor for homologous recombination self-renewing stem cells. So all this data I'm going to be showing you is from blood stem cells from sickle cell patients that we got from Stanford University. And so what we did is essentially we took the medical waste from sickle cell disease patients when they undergo transfusions, and we harvested the CD34s within the circulating red blood cell compartment. And we did a 34 max selection, and then we sorted stem cells and cultured them for 48 hours. And this is basically showing the picture of the stem cells that we use by flow cytometry. And this is what a typical experimental timeline looks like within our lab. So once we harvest the cells, we pre-stimulate them for 48 hours, and then we electroporate them with a chemically modified guide RNA complexed with the high-fidelity Cas9, and we transduce with our DNA donor molecule. So down here is showing what the donor molecule looks like. So this is the change we want to make. So this is the money maker. We're trying to make that change back to a wild-type beta globin. We make a few changes here to prevent Cas9 recutting, and then we make these 3 changes highlighted here to prevent premature crossover during homologous recombination. So after that process, we harvest the cells to assess the purity, potency, and viability of them, and then we transplant them into immunodeficient mice to assess their stem cell function to see if they can set up shop in the bone marrow and produce differentiated cells. So first here, showing on the left is the genotype outcomes following our platform. So basically, essentially, HBS is no change in the beta globin gene, so it's still HBS. We also get resulting indels, and indel stands for insertions and deletions. So it's a consequence of the double-strand break repair pathway. And then we also can get this E6V reversion. So this is what's showing HBA in about 50% of the alleles in this compartment, or in this gene. Then we differentiate these cells in vitro into red blood cells. And they can show quintessential red blood cell markers like CD235A or GPA, and they lose CD45. They also express high levels of the transferrin receptor, or CD71, and they lose CD34. After they're differentiated, we take these cells and we look for hemoglobin tetramers that are formed to assess and see if we were able to rescue the hemoglobin A. And shown here is this HPLC plot of cells that weren't targeted and cells that were targeted for homologous recombination. Of course, sickle cells express mostly hemoglobin S. When we use our technology, we're able to knock down hemoglobin S and rescue the hemoglobin A, so now they express functional hemoglobin. Another readout we wanted to look at to see if they had progenitor cell function. So we put them in a semi-solid media to see what kind of progenitors they can form, and it also gives us the ability to look at single-cell genotypes, because one of the things that happens in here is you develop cells that have a mixed chimerism, essentially. So here is showing the number of methylcellulose clones with a specific genotype. So HBS and HBS is essentially no change all the way to where a cell has change at both alleles. And you can see we have this mixed chimerism. But about 50% of the progenitor clones that we look at have a change from HBS to HBA. And this is a big point, because all of these genotypes essentially here would be functional in a patient when we transplant them. Some of them would have no change, like the HBS, HBS, and some would have a sickle beta thal phenotype. But over half of our events in cells that can form colonies have a change that would be beneficial for the patient. And then lastly, what we try to do is, again, we try to see if these cells can set up shop in the bone marrow and survive for 16 weeks and produce differentiated cells in an immunodeficient mouse. So on the left is showing cells that weren't targeted and showing the engraftment at a total CD45 within the mouse compartment. And on the right is showing our gene corrective cells that we call gene corrective HBV for sickle cell disease. And they can set up shop 16 weeks later. And really, really encouraging to us is that they can retain their homologous recombination within the bone marrow. And it's above this 20% chimerism that has been shown for allogeneic transplants. So it gives us hope that when we try this for a clinical trial, it has the potential to hopefully work for these patients. And so again, this gives us a lot of confidence that this can potentially be an investigational cell product. And so what we're trying to do here at Stanford is file an IND to the FDA in Q2 of 2019. So I just wanted to put this up here because I've been fortunate enough to be here in the beginning when the discovery was first made. And hopefully in 2019, we can start this phase 1/2 trial at Stanford through the CDCM. So right now, we're doing our long-term safety studies, our IND-enabling process development and feasibility studies. And so hopefully those are ongoing. So we're crossing our fingers that everything's going well. And then we can start this trial next year. And then with that, I'll take any questions. But I mean, everybody in this picture essentially helped. It takes a village, and I'm just here to relay the message. Thanks a lot.
The question about CRISPR correction for clinical use is how much off-target cutting there is, and how often do you see indels that might have deleterious effects?
So the first question, we're always below 1%. Luckily, we made a co-discovery with IDT that we found a high-fidelity Cas9 that really reduces these levels to less than 1%. But the second question...
1%, that's 1% of what? What's the denominator there?
1% of total alleles that we look at that locus. So if you look at an off-target site based on a bioinformatic, 1% of those alleles in the population would be cut. But most of them around 0.1. But the second question, I've never seen anybody ever generate data that an off-target could have a biological consequence in this field so far. That's just my personal opinion. Hopefully somebody can. So we're doing this long-term safety study to assess that question. So the FDA says we have to do this to show that hopefully we don't form any tumors. But my guess is that even if there were breaks that were deleterious, I don't think the mouse model would necessarily show us.
Yeah. I'm not trying to suggest that this is not a safe or a reasonable approach at all, but I think it's one of the general questions, given the difficulty of identifying the off-target hits. How often might you have a loss-of-function mutation, for example, in a tumor suppressor, that would increase the risks? Yeah,
I mean, we do an unbiased, non-bioinformatics, so like guide-seq, and then we do a circle-seq from Keith Young's lab, and then we also do a bioinformatic approach by basically making a list. And we do those three together and then pick the ones that we can actually see. I think the field is very far behind in terms of being able to see something that's actually functionally of a consequence. It's a good question, though.